what on May the 18th, we're going to have a talk about cancer and AT involving Malcolm Taylor, who you probably know does a lot of the diagnosis, Louise Isat, who's a consultant in clinical and cancer genetics, and Simon Bompkin, who's a pediatric oncologist. And the aim is that they're going to address some of the questions and more that you asked when we had the previous talk about cancer and 18 carriers. So what I wanted to do today is just give you a fairly brief overview, which is really a bit repetitious, of why ATM individuals are prone to cancer and carriers. And then more in preparation for the 18th, talk to you about what genotype-phenotype relationship is, what that means, types of mutational changes, the level of residual activity, and these couple of concepts about separation of function mutations and the concept of dominant mutations. And hopefully that will then really put you in a good position to understand and to be able to really get more out of the talk from these people and the questions. So I'm building on what I've told you before, but I'll try to just go over everything so that this talk stands alone. So your DNA is the code that makes everything that we need to make the human body. You can think about it as a instruction booklet. It's got these 4 building blocks, which you can think of in colors or as ATCG. And these 4 building blocks are arranged in a sequence, any sequence, on what we call a backbone, and they can interact with each other. So we get a ladder-like structure. This sequence of our DNA is unique to each individual, each one of us. And it's that sequence that gives the code to finally make what we call proteins. That code is quite complicated. It's got the code for make everything that we need to be, a cell in our head, a brain cell, a muscle cell, whatever cell you want. From that code, a more simplified code, which has the information to make just one protein or worker molecule, that's the first step. You make this more simple code. That's also built of 4 building blocks, but they're slightly different, but I've never gone into that difference. And there's just one strand, whereas these, you've got 2, what we call these 2 strands, and they kind of are arranged like a spiral to give you this DNA double helix, but this is just one strand. And then we have these proteins. They're the worker molecules of the cell. They do the tasks. They've got 20 building blocks, which are called amino acids, and the amino acids are added in a chain, entirely reflecting this chain here, or the sequence, but there's a lot of interactions between them. So for proteins, it's this 3-dimensional structure that's important, whereas for your DNA, it's really a long linear molecule, although it's divided into chromosomes, okay? So, and also the region on your DNA, which has got the information to make one protein, is called a gene. So we have Ataxia-Telangiectasia is the disorder. We have the ATM, which actually stands for Ataxia-Telangiectasia mutated, protein, and the region on the DNA which has the code for that protein is called the ATM gene. And similarly, we have the AOA1 protein and the AOA1 gene. Okay, now a mutation is a change in the sequence of these building blocks. That means that you'll also have a change in that sequence in the messenger RNA, and most importantly, that mutation it means the protein will be abnormal, or at least in the situation we're talking about disease. Sometimes it can prevent the protein being made, but sometimes the sequence will just change. It will just change the sequence of these building blocks here, and that will make an abnormal protein. So this is what we mean by mutations, change in the sequence of the DNA. And we're going to come back to this term gene in a minute, but that's the information here that determines this protein. Now, first, before I kind of talk about that a bit more, what's a cancer cell? A cancer cell are one of our own cells, but they've gained uncontrolled proliferation. They don't know when to obey the rules of society, really, when to stop growing, when to divide, when not to divide. And in order to achieve that state of uncontrolled proliferation, cancer cells acquire mutations. They have to constantly change. And normally about the certain number of mutations that we think have to be acquired for a normal human cell to gain uncontrolled proliferation. Here it's depicted as 4, but I think it may be more than that. But even once you've gained these 4 mutations and you can become a cancer cell, you still got to, for a cancer to grow and develop, it's got to constantly change. It may want to invade another part of your body, or it wants to have a bigger oxygen supply. And all of that requires continuous mutations. You can think of it like evolution that's fast-tracked. Many of these mutations will be harmful and not help the cancer cell at all, but evolution allows you to select for those beneficial mutations. So our own cells try to maintain the stability of our genome, that sequence of our building blocks. Cancer cells are addicted to genomic instability and the generation of mutations. They need it to adapt to all that changing environment. I've talked about previously how all the cells in our body get constantly damaged from external agents, but also from damage caused internally. But damage to the RNA and damage to the proteins isn't so important because it can be remade using that sequence on our DNA. So it's the DNA that's really important. We talked about all sorts of damages, but I said that a double strand break, i.e. a break in both of those strands of the ladder, ATM functions in helping these get repaired and restoring order if these two get damaged, both in a similar or overlapping site. Single strand breaks don't require ATM, but AOA1, Ataxin, which is the protein defective in AOA1, is involved in repairing both those lesions and these lesions. ATM doesn't actually do the repair, but I've said before, think of it as a policeman that regulates a response. And because of that, it affects many endpoints. I'm not going through all of those today, but what's relevant for cancer is that it promotes the accurate repair of those two, when you get a double strand break, a break in both of those strands. And that restricts mutations arising. So it's anti-mutagenic by and large. But it also helps to remove cells when they're damaged and stop them proliferating. So as a policeman, ATM helps to maintain that genomic stability, and cancer cells need to adapt and change. So I kind of found this on the web. ATM is called a tumor suppressor gene because it's suppressing cancers arising, tumors. It puts the brakes or a stop signal on a tumor. So you can think of this as a tumor. This is a tumor suppressor, which is stopping this moving along towards cancer. And this is called an oncogene, which is pro-cancer. Many of those proteins that repair our DNA damage are tumor suppressor genes because they restrict mutations arising. But in fact, ATM protects a bit more also about survival. It helps you survive, not just not get mutations. So ATM is not quite such a strong tumor suppressor gene, but nevertheless, it is one. And cancers often want to mutate or change their tumor suppressor genes because they're suppressing cancers developing. And so some cancers have actually changed or decreased ATM activity. That's true for chronic lymphoid leukemia, is true for breast cancers. So losing ATM is a step enhancing cancer formation and progressing. So that's why individuals who have lost ATM are more prone to getting cancers. Just one other thing before I finish this introduction is to say ATM plays this function particularly during the development of the immune response. So the development of the immune response requires us to form T and B cells, which are our antibodies, they want to create genetic diversity because we want to recognize all the different foreign bodies to which we're exposed, like coronavirus. And to do that, the cell has to create genetic diversity. And so what the cell does during this creation of your immune response, generation of your immune response, is to put in double-strand breaks and then deliberately rejoin them inaccurately. And that process does work in the absence of ATM. But what ATM particularly does is to police it and get rid of bad immune cells that have the opportunity to become cancer cells. And because of that, individuals are more prone to lymphoid tumors. It's sort of an indirect consequence of the role of ATM in the immune response. They do, as well as that, you do get other tumors in other sites as well. The other reason why the lymphoid tumors, I think, tend to be more prevalent in AT individuals is because they tend to develop more quickly. But there are other individuals who are cancer prone who don't show this predisposition to lymphoid tumors. So that this is an additional aspect here, is the role of ATM in the generation of the immune response. Okay, so that's really the sort of, if you like, the more theoretical aspect of why AT individuals might be getting cancer. And I've gone through that quite quickly because it's a summary of what I've told you before. But what I want to try and talk about now is some of what I think might come out in this cancer talk. So the first thing is, what do we mean by genotype-phenotype relationship? Now, your genotype is the nature of your genes, and that's effectively determined by the sequence of those 4 building blocks, which I've told you. The phenotype is the observable characteristics or traits, and that's determined by the protein. Now, the protein is determined by the sequence of these building blocks, but there's a slightly different relationship there. So the relationship, genotype, phenotype, asks how does the nature of the mutation affect the observable characteristics? Just to bring out what we're not going to talk about initially. So each of us has a unique DNA sequence, and we differ in many genes. We don't all necessarily have a gene that's so severe or change that's so severe it'll cause something like AT, but we differ in many different ways. And sometimes we have changes that make some of our pathways just very slightly different to others. Particularly immune responses, we can be very different. So some of the variation in AT features, and particularly including cancer, will be due to those differences rather than differences to do with the ATM gene itself. And as an example, there is an ATM patient who has no ATM, but she had no ataxia until being an adult. And that difference was due to some other aspect of her genotype. The other issue to remember is cancer is stochastic. Even some of us without with normal ATM will get cancer, some won't. So there's going to be differences between individuals. But what we want to focus on here is how the differences in the ATM gene sequence, the nature of the mutations in your children can affect the differences in cancer. And that's what we mean by the ATM genotype-phenotype relationship. Okay. Now, just to go back to this relationship of DNA to RNA to protein, making the protein, we're going to focus for a minute on this step 3. And this involves a protein factory. This is called translation. The protein factory. And you may remember in a past talk, I talked to you about how the question is, how do you get a code just made up of a sequence of 4 building blocks, that's all. How do those 4 building blocks land up being able to enable you to put in one of 20 different amino acids in a sequence? And the way that was done, you may recall, is that I said this code is read off as a triplet, i.e. a group of 3. And if you think of, well, if you do sums and you've got 4 building blocks and you think of how many ways that can all be organized in a triplet, it comes to 64. But we've got 20 amino acids, 20 building blocks here. So there's some redundancy, but the important thing is it's this you're reading off in this group of 3 called the triplet code. And then critical to the protein function is that 3-dimensional structure, which is here, going on to your 3-dimensional structure there. And that's based on the order of these amino acids, and each one of these will encode for a specific amino acid. So we'll come back to that in a minute. But then for now, let's just talk about this earlier step here. And this is making your messenger RNA from your DNA. So the first step there is you make an exact copy, and this is called pre-messenger RNA. It's not shown here. You make an exact copy of the sequence of those building blocks here into RNA. But the second step is to cut and splice to remove bits of this DNA. And you may remember me talking about this. So this is your DNA sequence along here, the sequence of those building blocks on this molecule here. And to make this messenger RNA, there are bits in here, in that sequence, which you're not going to use. And that's these little ones in small letters here. And so to take the ones that you want to use, you cut and paste them together. And that's called splicing. And some of your families, some of your mutations arise when this cut and putting together is not done accurately. And you have about 60 of these cut and splice things in the ATM gene. And they're called splicing mutations. And they, if they're erroneous, we'll talk about what that can cause in a minute. And then this finally put together as your messenger RNA is going to be read off in this group of 3 to put in these different building blocks of the proteins. Okay. No, I'll tell you that next slide. Okay. So now we're just going to think again about what types of mutations are there. You can have what's called a missense mutation, where literally one of these mutations here just gets changed to something else. In this case, a T has been changed to a C. So instead of reading CTC, which will put in a certain amino acid, it's reading CCC, which will put in a different amino acid. That's at that one point. And that is called a missense mutation. It's just a change in one of those building blocks, and it will cause a single amino acid to be different. We talked about insertion or deletion. Sometimes you can add in a few building blocks here. Now, if it's 1 or 2, that's a problem, because your little triplet code is going to be always out, because instead of reading here, you might be reading here. If it's an exact group of 3, it's not quite so bad, because you may have one more in there that you didn't need to have, or 2 or 3 more that you didn't mean to have, but everything downstream will be all right. But if you put in 1 or 2, insert or delete 1 or 2 of those building blocks, you're going to be out of frame, and that's serious. And then we talked about a change in the splicing, which is where these points where you cut and put them together again. And that's a hard task for the cell. And sometimes the mutation will change how you do that. So instead of, for instance, splicing from there to there, you might splice from there to there. An important thing about splicing mutations is that sometimes, not usually I would say, but sometimes, although you may be doing it mainly wrongly, you do it correctly a little bit at a very low frequency. We'll come back to that. So now, what type of mutations can we think about in terms of changing the genotype, phenotype? So we can have what we call null mutations, which we're not making any residual protein. Nothing is made. That's one class, and we'll come back to that in a minute. You can have some classes where you do make a little bit of residual protein, and it's got the normal sequence or the normal function. And that can occur, as I said, when you have splice site mutations, because sometimes you do that splicing just correctly. Or you can have some residual protein, but the sequence is completely abnormal. Well, not completely, but is abnormal. And that may be just a very localized one little change that's abnormal, or it could be a lot of it's abnormal. And that commonly occurs with a missense mutation, where you're just putting in one of those building blocks that's wrong. Now, null mutations mean that you're not making any protein. So all patients with null mutations are going to be, should have a similar phenotype. That means a similar age of onset. We know it's not identical. There's something stochastic about when you're going to get ataxia, but at a sort of absolute level, any patient who's got no protein should show similar features overall to another protein, and any differences will be due to the differences in those individuals. The only thing I would say is you have to remember that you've got 2 copies of that ATM gene, and one may be null, but the other one may have some residual activity. So you'll always be determined, or the phenotype will be determined usually by the one with some residual activity. So just one non-allele may be distinct to having one non-allele and one allele with some residual protein. Okay, so now we're going to talk about the situation where you have residual protein made with normal sequence. And at the extreme, in a way, that is a carrier. So a carrier has 50% of the wild type protein. Normal, wild type just means normal, but has no ataxia, no immune deficiency, but some cancer predisposition. So although just 50% of the activity still gives you some cancer predisposition. AT variants can be very different, but let's initially think about a person who has 10% of the normal level of activity, but that level of activity, that type of activity, the sequence is completely normal. So it's just much lower. That won't be all variants, because some variants may have the protein may be abnormal. But generally, variants of this sort do exist. They have later onset ataxia, minor immunodeficiency, but actually their cancer predisposition can be quite marked. They tend to have fewer childhood cancers, but increased cancers in older individuals. So to some degree, the greater, or it's not greater cancer predisposition, but the cancer predisposition you're seeing is just because they're living longer, and you're seeing them in older individuals. But if you think about all of that together, some residual activity has a pretty good effect at delaying the onset of the ataxia. It has a significant impact on preventing immunodeficiency. But the protection against cancer is complicated, and it's definitely not 100%. And even a carrier does tend to still have some predisposition to cancer. So the different phenotypes, i.e. different traits caused by ATM loss, may differ in their demand for the level of residual activity. And cancer in general is quite demanding. It wants some, even if you're a carrier, you still have some cancer predisposition. Now, we're now going to talk about the residual activity with abnormal function. And the first case I'm going to talk about is the impact of a missense mutation. So here, the important, sorry, just stopped. I got some phone call coming through to me. So the way a protein folds is critical to the function. And a missense mutation can have just a very local impact, or it may have a much broader impact. And some proteins can have multiple functions, and there are you can get what we call separation of function mutations, which means that you may affect one part of the protein, but not the other part of the protein. And now for ATM, its kinase activity, that the ability to tag a protein and change it, is really the only known function. But there are ways, and I don't want to go into that, into how you may affect one aspect of the protein without affecting another aspect of the protein. And that's called separation of function. And for cancer, that can be important. And at the extreme, although this isn't true for AT, there's something called AT-like disorder. And this has mutations in MR11. And MR11 is involved in activating ATM. So we have some ATLD individuals within AT society. And MR11 can activate ATM. So some of the mutations cause something which is called AT-like disorder. And it's a very mild form of AT. But there are other mutations in this same protein, same gene, which don't look at all like AT. They don't have progressive ataxia. They have microcephaly small heads, completely different area of brain where there's a problem. Very, very distinct, but it's mutated in the same protein, the same gene, and they're called separation of function mutations. And there is some aspects that you can get that with AT. Some mutations may affect one bit of the protein rather than another bit of the protein. So just to kind of show you a picture of how putting in one little building block can affect the function of a protein, I chose this figure. Well, I've got two figures. This one here. This isn't ATM, but this is a protein called ligase 4. And basically, these are those building blocks called amino acids here. And here you've got something called ATP, which is a tiny little protein, but it binds to this particular protein and it gives the energy. It's not really a protein, it's a chemical group, but it gives it the energy. So this binding of ATP is critical. And the way it binds this structure is involving these these ARGs called arginine. These are amino acids. So it binds here and you get little bonds forming between these things. And it's all chemical reactions. Now, in this individual, who was a young boy, he was 14, he developed leukemia and was given radiotherapy for his leukemia and died. And it was all due to this little change here. So instead of having this amino acid, he had this one, and it's bigger and bulkier. And this ATP wasn't able, therefore, to form these little bonds. And that prevented the function of this protein and landed up making the little boy prone to leukemia as well as then sensitive to radiation. So here, this is more to do with the bulk. This is bulkier, bigger than this little amino acid. But often it can be charge and interactions between other components here. The other example is, and this is the ATM, there was a little mutation. So this is normal, and this is where there's a mutation, and you can see it's changed exactly how this protein is looking around here. And that can sometimes have bigger impacts as well. It's made it look a little bit different down here as well, up there. So sometimes the impacts can be quite local, but sometimes they can be quite big. And sometimes if they're in a really critical part of the protein, the cell recognizes that protein's not normal and degrades it. So just one single little building block can be quite minor. Sometimes the change is almost so you don't know it, but sometimes it can be quite major and completely destroy the protein. So the last example I want to talk to you about is, no, one more. So two examples I want to talk to you about. This is where you've got a protein with abnormal function. And occasionally the presence of ATM, rather than its function, can stop other proteins acting. And the example I'll give you here is, so we know mice completely lacking ATM have been generated, and those mice, they're viable, but they don't generate it, they don't develop ataxia. So they're not completely exactly the same as humans, but humans without ATM are viable. Now, in the laboratory, a mouse has been made which expresses ATM completely normally, but it's not able to function in that tagging activity that I talked about. So it's got no function, can't do its task. And that mouse is embryonic lethal. So the presence of abnormal ATM, i.e. without kinase activity, is different, and in fact more lethal than not having any ATM at all. And one way I think you can think about it, although there's several different possible explanations, is that you can have something else may be able to bind here if you don't have ATM at all. And there is a protein called ATR, and there can be some overlapping function. So this can do a little bit of what happens if you don't have ATM. But if you've got ATM bound there, but it's not doing its job, you're worse off. So in this particular case, a little ATM is worse than no ATM. I'm not aware that there's patients that are exactly like that, but there may be, and there is some evidence of individuals with AT who who can be worse off than not if they didn't have any ATM. And then related to that really is this final topic I wanted to talk about, which is called dominant negative. And just before I do that, I just want to remind you that we've talked about ATM being a recessive disorder. What that means is you have a mutation, you've got two copies of your ATM chromosome and your gene on the chromosome. And the two parents are carriers, they've each got one copy mutated. And then when you have children, you'll have a bad copy with a bad copy with a good copy, carrier, good copy with a bad copy, and two good copies. And this is called recessive because these individuals don't show AT. So bear that in mind. And now we're going to think, so AT is called autosomal because that means it's on chromosome 11. It's not one of the sex chromosomes, which means you always have two copies of it. And it's called recessive because to have the major phenotype, i.e. progressive ataxia, you need to lose both copies. And there are sometimes dominant diseases where you only have to mutate one copy. And there are some ataxias that are like that as well. You may have another copy, but if you only have one, if you have one bad copy, that's enough to give you the disorder. Now, there can be mutations that are called dominant negative mutations, and they basically affect the function of the normal protein. So if you think of a null mutation with a leaky mutation, you won't see anything here, but you've got a little bit of activity. So you get slower ataxia than a normal AT individual. It's slower onset. But if you have a dominant negative mutation rather than a null, i.e. you do make the protein, but it can stop the other protein working, that together with the leaky mutation, you may be worse off and you have ataxia like a normal AT individual. And even this dominant negative in a carrier with a normal copy, you may still have some ataxia. So the presence of this guy is actually worse than having no protein. And I tried to give you an example of how to think about that, one way in which that can arise, just to help you. But if you find this difficult, just ignore what I'm saying here. But ATM, the way it works is that you have, it forms what we call a dimer. It binds to itself and is without damage, it's inactive. But when you get that damage there, that double strand break in your DNA, it becomes separate. It forms what we call monomers and is now active and is able to tag those proteins and set up the response. So here I've depicted normal protein from one of the chromosomes, from the other chromosome, binding to itself. But when you get the presence of damage, it can separate, become what we call a monomer, and you've got 4 times the activity because each one of these will be active. In fact, you'll have multiple molecules in the cell, but I'm just trying to show you what happens here with a few. Now, in this particular patient I've shown, you've got one null allele, so you haven't got any activity here. And you've got another copy of the ATM, which is, I've said, half the activity of the normal one. So they can form together, they can become separated, and each one of these will have half the activity. So that gives you nothing. This gives you half. So you'll have 1 times the total level of activity here. Now, if you've got one of these dominant negative mutations, bad guy, in this particular scenario here, you can have a dominant negative binding to itself, but it can't ever become active. So it's not active there, but this binds to itself, does what you've got there. So you've still got some active protein. That's fine. But what you've got to remember is that in a cell, you've got a mixture of these proteins. And sometimes one of these will bind to one of those. And if that can stop one of these being active, you'll then have no activity because one of these little red guys can stop the other guy being active. So in fact, you're worse off having one of these than having no protein. And that is called a dominant negative. And these type of mutations are not so common for affecting ataxia, but they can be there for cancer. And that Malcolm may talk about that. Okay, so to summarize, I've told you what we mean by genotype, phenotype, how a mutation affects the characteristics. Normally, the phenotype, i.e. the characteristic trait, is dictated by any residual activity in one or both copies of the protein. Occasionally, a mutant protein can block another process, so you can be slightly worse off than if you had no protein. And sometimes you can have something called a dominant mutation or dominant negative mutation that can counteract any residual, the good protein, any residual activity of the protein. And that is not something that you want. Okay, so that's just trying to tell you the different types of mutations you can have and how they can affect what we're actually seeing. And I think so. I'll stop this.